Hello and welcome to my channel. Today's lecture is about the cerebellum, so let's begin. The cerebellum, they call it the small brain. It lies in the posterior cranial fossa underneath the tentorium cerebelli. So remember, what occupies the anterior cranial fossa is the frontal lobe of the brain. The middle cranial fossa is the temporal lobe of the brain, and the posterior cranial fossa, not the occipital lobe, but the cerebellum. Remember that. It's composed of two hemispheres, as you can see here, connected by a vermis. This is the vermis. Vermis means worm-like. It has three lobes and three fissures, three peduncles, and cortex has three layers, just to be able to remember. The lobes are anterior, posterior, and flocculonodular lobe. Now, from the superior surface, this is the anterior lobe. The posterior lobe is this part and all this part. The flocculonodular lobe is this part only. The flocculonodular that has the flocculus and the nodule. Flocculonodular lobe. Fissures, we have the primary fissure. This is the primary fissure here at the superior surface. Horizontal fissure, it's at the edge of the cerebellum, separating the superior and inferior surfaces, and the uvulonodular fissure that separates the flocculonodular lobe from the rest of the cerebellum. The peduncles through which the cerebellum connects with the brainstem is the superior cerebellar peduncle. It goes to the midbrain, middle cerebellar peduncle to the pons, and the inferior cerebellar peduncle to the medulla oblongata. It's covered by tentorium cerebelli, as mentioned before, and forms the roof of the fourth ventricle. We saw that in sections of the fourth ventricle when we studied the fourth ventricle. The cerebellar cortex has three layers: outer molecular, the Purkinje cell layer, and the granule cell layer. So outer molecular, Purkinje, and granule. The molecular layer is this layer, has a collection of neurons and neuroglia, like stellate neurons and basket neurons. But the most important is to remember that it has the dendritic tree of the Purkinje cells. See, this is the dendritic tree of the Purkinje cell. These are Purkinje cells. And the parallel fibers of the granule cells. Parallel fibers. Parallel means they run parallel to the surface. Purkinje, the granule cells actually form parallel fibers in the molecular layer, or between the molecular and the Purkinje layer, and they anastomose with the Purkinje cell layer. So remember, the dendritic tree of the Purkinje cell is perpendicular to the surface, while the parallel fibers of the granule cells are parallel to the surface, and a neuroglia to support the neurons present. Purkinje cell layer, the Purkinje cell is the functional unit of the cerebellar cortex. Means that all other cells act on the Purkinje either to stimulate or inhibit the Purkinje cells, and it's the balance between the stimulation and inhibition that determines the functions of the cerebellum. Dendrites from the dendritic trees perpendicular to the long axis of the cortex anastomose with the parallel fibers of the granule cell layer, and the axons of the Purkinje cells go to vestibular nuclei and cerebellar nuclei. Now, because it goes to the vestibular nuclei, these are the only cerebellar cells that project to outside of the cerebellum. And actually, the vestibular nuclei considered the deep cerebellar nucleus of the Purkinje cells. So they project by their axons to vestibular nuclei and the cerebellar nuclei, which we study later. Purkinje cells are inhibitory neurons and receives excitatory input from the granular cells and inhibitory input from other cells. So they receive excitatory input from granule cells or granular cells and inhibitory input from other cells. Granular cells and other control the action of Purkinje cells. Okay? The granule cell layer contains granule cells, Golgi cells, and neuroglia. The granule cells, the cells themselves are not granular, but because they are small in size, they give the layer the granular appearance. They have small dark nuclei, and the dendrites synapse with others. Mossy fibers we'll get to this later on in this lecture. Mossy fibers are any other fibers in the cerebellum entering the cerebellum. They synapse with the granule cells by their claw-like ends. Golgi cells, they have dendrites to molecular layer, and axons synapse mainly with the granule cells, and neuroglia cells, of course, to support these cells. So to summarize, Purkinje cells are inhibitory, as mentioned, and give projection to outside the cerebellum to vestibular nuclei, while the granule cells are excitatory and interneurons. Other cells like the basket and stellate are inhibitory and interneurons. So the purpose of the cerebellar circuits is to modify or control the action of Purkinje cell inhibitory activity, either increase their inhibitory activity or decrease their inhibitory activity. White matter of the cerebellum, it contains 4 deep cerebellar nuclei buried in the white matter, the same principle of the basal nuclei in the cerebrum. The fibers of the white matter are intrinsic fibers that connect the different regions of the cerebellum. Afferent fibers, they enter the cerebellum through the inferior cerebellar peduncle, the middle cerebellar peduncle, and the ventral spinocerebellar enters through the superior cerebellar peduncle, although this is questionable. Some consider the fibers of the ventral spinocerebellar go through the inferior cerebellar peduncle as well. So there are no afferents in the superior cerebellar peduncle. But anyway, the fibers entering are the afferent fibers. And there are some fibers called the climbing fibers. They are fibers that distant to synapse with the dendrites of Purkinje. Any incoming fibers to synapse with the dendrites of Purkinje are called climbing fibers. They originate in the inferior olivary nucleus in the medulla. So it's a connection between the climbing fibers, connection between the inferior olivary nucleus and the cerebellum, to be precise, to Purkinje cells, and collaterals from climbing fibers to deep. They send collaterals to deep cerebellar nuclei as a feedback loop. What else? We have mossy fibers. All other tracts entering the cerebellum are called mossy fibers. They synapse with the granule cells, then through the parallel fibers of the granule cells to the dendrites of Purkinje. So more or less they affect the function of the Purkinje. They give collaterals to deep cerebellar nuclei as a feedback and stimulate Purkinje cells. Parallel fibers, why they are parallel to anastomose with as many as Purkinje cells as possible. So parallel fibers stimulates thousands of Purkinje cells. Efferent fibers, axons of cerebellar nuclei, and some axons of Purkinje cells to vestibular nuclei. So the fibers leaving the cerebellum are the axons of cerebellar nuclei. We'll get to them now, and some axons of the Purkinje cells to vestibular nuclei. The deep cerebellar nuclei, they are present in pairs, right and left. From lateral to medial, they are dentate. The biggest one is the dentate. Emboliformis and globose together, and the fastigial nuclei in the middle. Emboliform and globose, they are called together, they are called the nucleus interpositus, or the interposed nucleus. And they form the main output of the cerebellum together with axons of Purkinje to vestibular nuclei. Now the dentate nucleus, the emboliform and the globose output through the superior cerebellar peduncle, and the fastigial nucleus output through the inferior cerebellar peduncle. Remember, these are efferent fibers, not afferent fibers. But the afferents to the deep nuclei, they come from the Purkinje cells and are inhibitory. From collaterals of the climbing and fibers coming from the inferior olivary nucleus and are excitatory, and from collaterals of mossy fibers are excitatory as well. And superior cerebellar peduncle, inferior cerebellar peduncle contain both afferent and efferent outflow. Remember that middle cerebellar peduncle, which is composed of the transverse pontine fibers. Remember, the fibers synapsing on the pontine nuclei and forms the middle cerebellar peduncle of the opposite side. They contain only afferent fibers. I hope you got the maximum benefit from this lecture, and thank you very much for watching.